Today we're going to cover the cell membrane. The center of the cell membrane is a water-loving section of the molecule, hydrophilic, liking water, right, which is the phosphorus. The ends of the membrane are hydrophobic. That will be the lipid end of the membrane. This constitutes the whole phospholipid bilayer of the cell membrane.
Yeah. I'm Anna Purvis, and I did a vacuole. The vacuole maintains fluids, removes waste, and stores food, and provides and maintains cellular structure. Vacuoles are little pockets in the cytoplasm of a cell where a cell stores food. So they store food and water, like I told you. A vacuole is a membrane-bound sac that plays roles in intracellular digestion and releases of cellular waste products. This, as you can see, is a vacuole. This is the waste, and it's not letting the waste in, it's spitting the waste out, as you can see, it's off of the vacuole. And it's storing water, which is the blue, and then the food, which is the white. Yeah. And it basically is the store cell for the cell.
And it brings it out to the cell membrane and brings the waste out of the cell like that?
Yeah.
Does it also bring things into the cell?
Water and good food.
Nutrients?
Yeah.
I'm Tiffany Mustafa, and I did the nucleus. The nucleus is membrane-enclosed organelle that's found in eukaryotic cells. It pretty much carries the cell's DNA and the chromosomes. It's made up using the nuclear envelope, a double membrane that encloses the entire organelle and separates it from the cellular cytoplasm and the nuclear lamina, which is like a meshwork within the nucleus that adds mechanical support. Because the nuclear membrane is impermeable to most molecules, nuclear pores are required to allow movement of the molecules across the envelope. And the nucleus works together with the ribosomes, the endoplasmic reticulum, and the nucleolus to make protein.
The nucleolus is located in the nucleus and plays a role in protein synthesis. It is actually a suborgan. It is in the nucleic membrane and forms subunits, which together form ribosomes. The ribosomes create proteins from amino acids and send proteins to the nucleus and through the ER back to the nucleus. Those proteins are used for a variety of things and are heavily situated throughout the body. The nucleolus also produces about 50% of the body's RNA, ribonucleic acid, and no membrane separates the nucleus from the nucleoplasm. It is a necessary component in protein synthesis and is the sole creator of ribosomes.
I'm Katie Colvin, and I'm doing the ribosomes. Ribosomes are components of cells that make proteins from each and every amino acid.
And they're found in
They're
the...
found in the nucleus. It's usually referred to as the central dogma. The DNA sequences and genes are copied to a messenger RNA. Ribosomes read the information that in the RNA and use it to create proteins known as translations. The ribosome translates the genetic info from RNA into proteins. They are also made up of RNAs and proteins, but the general thing they do is make them. They're divided into subsections, which are also divided into smaller subsections. And in bacteria, archaea, and eukaryotic, ribosomes have greatly different structures and RNA sequences. These differences allow some, but not all, antibiotic to kill bacteria by stopping their ribosomes, leaving human ribosomes unaffected. Different types of ribosomes can be different sizes. Prokaryotic ribosomes can be about 20 nms in diameter. Eukaryotic ribosomes are around 25 and 30 nms in diameter.
Nanometers, right? 10 to the minus 9th of a meter.
Yeah, okay. Yes.
Alright, I'm Kilo Hoops with the endoplasmic reticulum, and the main thing that this does is it connects to the nucleus, helps with the Golgi body and everything to connect with tubules, and helps the ribosomes produce proteins and send them throughout the cell. So I got like little ribosomes and Golgi bodies working in my endoplasmic reticulum. And the rough endoplasmic reticulum synthesizes proteins, and smooth endoplasmic reticulum. The rough endoplasmic reticulum synthesizes proteins, and smooth endoplasmic reticulum synthesizes lipids and steroids, and it attaches receptors on cell membrane proteins. So over here somewhere.
Here's a cell membrane, right?
Yeah, so I would connect the tubules that are in there into here, and I'd help synthesize lipids and stuff like that.
Cool. Microtubules,
So
right?
yeah, the endoplasmic reticulum.
The old Golgi apparatus is an organelle that modifies, sorts, and packages proteins into sacs called vesicles. The apparatus, the vesicles that confuse with the cell's membrane to release proteins into the environment outside the cell. This thing is located in all eukaryotic cells.
Eukaryotic.
Cells, this thing.
Brings it out to the cell membrane, right?
Yeah.
And then out of the cell, right?
And then all the way up there.
Cool.
It's because it's the Rejected Society.
It's society.
Amy McCann, and I'm doing cytoskeleton. Cytoskeleton is contained in the cytoplasm and is made out of proteins. Cytoskeleton is present in all cells, though it was once thought to only be in eukaryote cells, though research found them in the prokaryote. They are used in the movement of vesicles and organelles, as well as cell division. There are cytoskeleton filaments, which have the microfilaments, the intermediate filaments in the microtubule.
It's okay.
And cytoskeleton provides structure and shape to the skull, and the cytoskeleton elements interact extensively with the cytoskeleton membrane.
Cell membrane, right? Okay.
Oh, my name is Devonte Harris, and I'm doing the rough endoplasmic reticulum. The surface of the rough endoplasmic reticulum is studded with protein manufacturing ribosomes, giving it a rough appearance. However, the ribosomes bound to the rough at any one time are not stable and are not a part of this organelle structure, as ribosomes are constantly being bound and released from the membrane. A ribosome only binds to the endoplasmic reticulum once it begins to synthesize a protein destined for the secretory pathway.
Aaron Barajas, and I did the cell wall. The plant cell wall is a remarkable structure. It provides the most efficient difference between plant cells and other eukaryotic cells.
Eukaryotic.
Eukaryotic cells. The cell wall is rigid and gives plant cells a much defined shape. While most cells have an outer membrane, none is comparable in strength to the plant cell wall. The cell wall is the reason for the difference between plant and animal cell functions. Because the plant has evolved this rigid structure, they have lost the opportunity to develop nervous systems, immune systems, and most importantly, mobility. The cell wall is composed of cellulose fiber, polysaccharides, and proteins. In new cells, the cell wall is thin and not very rigid. This allows the young cell to grow. This first cell wall of these growing cells is called the primary cell wall. When the cell wall is fully grown, it may retain its primary wall, sometimes thickening, or may deposit new layers of a different material called a secondary cell.
I'm Olivia Warren, and I did Golgi apparatus. Golgi apparatus is a collection of vessels and folded membranes in the cell. It stores and transports the protein to the endoplasmic reticulum. The Golgi complex contains a lot of vessels. The vessels are used to send molecules to the cellular membrane. The main organelle that the Golgi apparatus interacts with is the endoplasmic reticulum, because in order to make new proteins, they're packed in the rough endoplasmic reticulum and then sent to me. So basically, the Golgi apparatus is responsible for transporting all the cells to the endoplasmic reticulum.
On.
In the nucleus of each cell, the DNA molecule is packed into threads-like structures called chromosomes. Each chromosome is made up of DNA tightly coiled, many times around proteins called histones and that support its structure. Chromosomes are not visible in the cell's nucleus, not even under a microscope. When the cell is not dividing, however, the DNA that makes up chromosomes become more tightly packed during cell division and then visible under a microscope. Most of what researchers know about chromosomes was learned by observing chromosomes during cell division. Each chromosome has a construction point called the centromere, which divides the chromosome into two sections or arms. The short arm of the chromosome is labeled the P arm, the long arm of the chromosome is labeled the Q arm. The location of the centromere on each chromosome gives the chromosome its characteristic shape and can be used to help describe the location of specific genes, its life in a cell.
My name's Kayla Cruz, and I'm doing ribosomes. In a cell, ribosomes are located in two regions of the cytoplasm. Some ribosomes are found scattered in cytoplasm, while others are attached to endoplasmic reticulum. Accordingly, endoplasmic reticulum, which membranes are bound with ribosomes, are called rough endoplasmic reticulum. Both the free ribosomes and bound ribosomes have similar structure and are responsible for production. Ribosomes and bound ribosomes have similar structure and responsible for production of proteins.
Thank you.
I'm Janine Stafford. The main structure making up the nucleus are the nuclear envelope, a double membrane that encloses the entire organelle and separates its contents from the cellular cytoplasm, and the nuclear lamina. Meshwork within the nucleus that adds mechanical support that, much like the cytoskeleton, supports the cell as a whole. Because the nuclear membrane is impermeable to most molecules, nuclear pores are required to allow movement of molecules across the envelope. Those are nuclear pores.
And they allow things in and out of the nucleus, correct?
Yes.
Thank you. Go.
The nucleus is defined as a genetically determined element. A NOR is composed of tandem repeats of RNA genes, which can be found in several different chromosomes. The nucleus is a participant in the ribosome assembly. The assembly not only involves the RNA, but ribosomal proteins as well. The genes including these proteins are transcribed by Pol2 in the neuroplasm, but a conventional pathway of protein synthesis, a process nuclear export of mature and NRA and translation of chromosomatic ribosomes. The mature R proteins are then reported back into the nucleus and finally into the nucleolus.